Okay, everybody, good morning and welcome to our lab today on membranes. So, if you go to your ebook, you'll find that we're doing exercise 10 cellular membranes. We're going to be looking at the structural integrity of the plasma membrane on the cell. This is not only found on eukaryotic cells, this is also on prokaryotic cells and even in plants. Cell membranes are very important because they are the selectively permeable barrier that selects and allows for only useful substance to go through to the inside of the cell and prevents any harmful pathogenic infectious microbes, any toxic waste material from entering into the cell. So, in order to understand how the cell membrane works, we have to break down its structure. How does it look like mechanically? What does the cell membrane look like? And then we'll be able to find out the function. So, if you look at the plasma membrane, you really need to know something that's very important. This plasma membrane is like a universe. The plasma membrane is like a universe in two things. Well, if we look at the cell, let's exactly look at where is the plasma membrane. So we have the cell. The cell has three compartments. The first compartment is the cell membrane or the cell plasma membrane, and we have the cytoplasm, the cytoplasm. And what is the last compartment besides the cell membrane and the cytoplasm? What's the last compartment? Besides the cell plasma membrane, besides the cell plasma membrane, we have a cytoplasm. And what's the last compartment in the cell besides the cell membrane and the cytoplasm? Yes. So the organelles are part of the cytoplasm. You see, the cytoplasm is like a swimming pool. Every swimming pool has water and rubber duckies floating on top of it. So the cell, the water-like structure is the cytosol. The cytosol. That's the fluid part in the swimming pool. The organelles are the rubber duckies that are floating on the cytosol. These are the organelles. Those organelles are like your mitochondria, your lysosomes, your peroxisomes, your endosomes, your proteasomes, your endoplasmic reticulum, and so on. So those are the organelles, those are the rubber duckies floating on top of the cytosol. What's the last compartment? Yes, please. Yeah, the nucleus. So this nucleus is the home of the DNA, where we have this huge polymer of nucleic acids that is divided into genes. The nucleus is like the pharmacy. All of us show up to the pharmacy. The pharmacy has all the medications out there that you can think of, but you're only getting what you have on your prescription. So same thing. The nucleus has all the genes, but not all the genes are being transcribed. Only the ones for which you have the ability to code, which is transcription. You know, transcription, you transcribe DNA into mRNA. So this transcription is started with the polymerase enzymes. These polymerases, they're going to transcribe the DNA in order to give you an mRNA that will be translated into proteins. So those three compartments are like universe. Why do I say universe? Because the universe, first of all, the universe is huge. If you look into space, the universe is huge and it's very diverse. It's very huge and very diverse. The universe has galaxies, the universe has stars, the universe has planets, the universe has the sun, meteoroids. It has everything, many diverse different things. And so do those all three. The plasma membrane has so many different things on it: phospholipids, glycolipids, lipoproteins, protein channels, cell adhesion molecules, cell adhesion proteins, leak channels, facilitated diffusion transporters. It has so many different things, just like the universe. So does the cytoplasm. The cytoplasm is like the universe also, because it has so many different things on it, so many rubber duckies, so many organelles, so many enzymes that are operating in there for the metabolism. And the nucleus is a universe also, because it has so much DNA and so many enzymes that are transcribing this DNA. So the universe is huge and diverse. And what else do you know about the universe? Is the universe stopped in its place, or it's moving? Is the universe moving, or is it dynamic or static? Is the universe dynamic, moving, or is it static? It just stopped in there and just like a pause. Yeah, it's dynamic. So the universe is dynamic. So are all three. Your plasma membrane is not like the fence outside your home, just standing in there. No, your plasma membrane is moving, and every second new channels are being made. You see those channels that I have here on the projector? These are aquaporins to allow for the movement of water. There are glucose transporters. For example, let's say you're fasting, you haven't had anything to eat, and you take a photo, a snapshot of your plasma membrane. Then you eat pancake. Then you take another snapshot after you had the meal. Will you think your plasma membrane gonna have the same quantity, the same number of glucose transporters after you ate than when you were fasting? Of course not. After you ate, your nucleus is going to be making a lot of more protein channels that will allow the passage of glucose to the inside of the cell, so we can metabolize this glucose that we ate and we make ATP out of it. So that shows you your plasma membrane is moving and dynamic. New channels are being made, new channels are being removed. There is a lot of, those phospholipids don't even stay on their places. They flip over. When you look at you see the plasma membrane, the phospholipids, those phospholipids, they flip sideways, they change place. They're not like standing like soldiers like this. No, they switch with one another. They switch places. They switch places horizontally. They switch places vertically. They flip over. So there's a lot of movement that's happening. So today we're just going to focus on here, this interesting universe, the plasma membrane that has phospholipids. Phospholipids. It has cholesterol. It has cholesterol. It has protein channels. It has cell adhesion molecules. What are cell adhesion molecules? What are those? From the name, cell adhesion molecules, CAMs, C-A-M, CAMs, cell adhesion. I'm going to ask you afterward. If you're not paying attention, you're going to lose a lot of points. CAMs. What are CAMs? Cell adhesion molecules. What are CAPs? C-A-P. CAPs and CAMs. CAMs, cell adhesion molecules. CAPs, cell adhesion proteins. So what do? They adhere the cells together. They stick the cells to one another. That's why you have a tissue. A tissue is like a collection of cells that are held stapled together. Those CAMs and CAPs, cell adhesion molecules, cell adhesion proteins, stick the cells to one another. Your cells, they don't sit like how you are sitting now in the tissue. You guys have gaps between you. Cells sit like how you're sitting in the subway, attached, like touching the person next to you. In fact, the cells are so tight together, they are stapled with these CAMs and with these CAPs, cell adhesion molecules and cell adhesion proteins. Where do we find the cell adhesion molecules? The CAMs. What do we find the cell adhesion proteins? CAPs. We find them on the plasma membrane. There's also protein channels. like tunnels that allow for the entry. Like, look at here. That's a channel that allows for the passage of water. These are known as aquaporins. Aquaporins. They allow for the passage of water. There's also glutes, Gluts. What are glutes? Glutes are glucose transporters. Transporters. Glucose transporters. They transport glucose from the bloodstream into the cell. You have lipoproteins on the plasma membrane, lipoproteins on the plasma membrane. You have glycoproteins on the plasma membrane. All this plasma membrane is rich, just like the universe. It has galaxies, constellations, it has moons, stars, meteoroids, sun. It has so many different things. So does your plasma membrane. Don't be overwhelmed. We're going to go over all of that, but you just remember that your plasma membrane is not just like a fence. It is so many different things together, and the different things, just like the universe, the sun is not standing in its place, the glucose is not just standing there in its place. They're all the stars, they're moving, so it's very dynamic. It's not a static compartment. So now let's look at the transport. So the cell membranes are barriers from the inner content of the cell and the environment outside. So different materials cross this membrane. So for example, when you have this plasma membrane, now you have created two environment. An extracellular environment, meaning you're outside the plasma membrane, and then you have an intracellular environment inside the cell. So inside the cell, we only want good things to come through in moderate amounts. We don't want too much sugar, so we pluck a lot of glucose transporters to allow for the entry of glucose to the inside of the cell. When we have enough glucose, we stop making those channels. You know, this plasma membrane is edited. You can edit it. Your nucleus edit it accordingly. If the nucleus wants sugar, it will put sugar channels on the surface to allow for the influx entry. So if you want to come inside, the plasma membrane, we make a channel for you, and this channel is going to allow the entry of the molecule of interest, and this is known as influx. While if we want to get rid of something and remove it, that is, we're going to efflux, removing material from the inside of the cell to the outside. Usually we efflux waste, we efflux metabolic waste, we efflux stressful substance. And sometimes this is why we did diffusion first last week, because what makes us influx, what makes us efflux, it's the concentration gradient. If you have a lot of sodium outside the cell, then you bet it's going to influx to the inside of the cell, because that's a concentration gradient for diffusion. Remember, we discussed that. Who's always influxing to the inside of the cell? Which electrolyte is always influxing to the inside of the cell? Sodium. Remember, sodium, there's too much sodium outside. So there's a concentration gradient. There is a diffusion pressure. Diffusion pressure that is favoring the movement, the influx of sodium from the outside to the inside. Who's effluxing from inside to the outside besides the waste? Potassium. Potassium, there is too much potassium on the inside. Potassium is the opposite of sodium. There's so much sodium on the outside. That's why when you open a door, it's going to flow to the inside. There's so much potassium on the inside. That's why if you open a channel, you will get a lot of potassium to efflux to the outside. Is that clear? This influx is based on a pressure, a diffusion pressure, a diffusion pressure. Who want to tell me what's their concept of diffusion pressure? What's diffusion pressure? You know, pressure, it means a tendency. There's a pressure to do something. There's a pressure. So there's so much outside, there's going to be a pressure for you to move inside. If there's too much potassium inside, there's so much is going to be a diffusion pressure to move outside. Osmotic pressure is different. Osmotic pressure is only for water. If there's a lot of solute next door, there's so much osmotic pressure to move, to water to move to the other side. Okay? So the pressure, you know they say peer pressure? So peer pressure is the same concept. There's so much drive. Pressure is a drive. Osmotic pressure, we said that osmotic, I said last week that osmosis, you know they say TM? When you see the word, when you see the two letters TM at the top of something, TM, trademark, that means this is something that is unique to like a certain trade. Osmosis is unique trademark to who only? Yes, please. To water. Nobody has osmosis besides water. If you have an alcohol and you put it in a solution, alcohol is not going to move from an area of a low solute to an area of a high solute. It doesn't care. It doesn't have osmosis. Only water will do that. Only water is going to go to a place where there's a high solute. It's like if you have a place where there's a lot of crime, but you have like 500 doctors, they're not going to go in the crime and solve the crime. But if there's 500 police officers, they're going to do the osmosis because that's their job. That's what they go and do. They dissolve things. So the water is the only one who dissolves. So you need to remember that this plasma membrane is selectively looking at who's allowed to come in, who should go out based on the metabolic need for the cell survival, okay, in order for the cell to survive. I hope I made it clear by now that this plasma membrane is a universe. It's moving, is not standing, and is rich in many different structures. Okay? Okay, so now let's look at the structure of the plasma membrane. That's in the book also. The plasma membrane is amphipathic. Amphipathic. What is that? Amphipathic. Amphipathic. You're going to see this word a lot. I'm pretty sure you guys went over it in lecture. So, what is amphipathic? Amphipathic is a word like hybrid. Hybrid. You know they say hybrid? Ever heard of the word hybrid? Hybrid. So, what is hybrid? They say hybridization. Hybridization. What is hybrid and what is hybridization, guys? Hybrid is like when you have two in one. You know, they say hybrid cars, they use gasoline fuel and electric fuel. You know, they do that. Now, when you look at amphipathic, amphipathic, this word is very important. Amphipathic means that we are two in one. Meaning, the plasma membrane has a red polar, red polar. It has a polar head, and it has two non-polar tails. One tail, two tail. So it has one head and two non-polar tails. And not only that, it's actually a double layer. So you have two heads, one on the top and one on the bottom. So you have one head on the bottom with its two tails and one head. So we call the top an upper leaflet. That's the upper leaflet. That's the upper leaflet. That's the upper leaflet. So where's the upper leaflet? The upper leaflet is this. This whole side is an upper leaflet. Upper leaflet. And this bottom side is known as the lower leaflet. Upper leaflet and a lower leaflet. So your plasma membrane has a double layer, an upper leaflet and a lower leaflet. Do you see it? There we go. This is here, or we'll turn off the lights. Okay, let's turn off the lights. Do you guys see? This is my upper leaflet with one polar head, two tails, and this is the bottom leaflet with a polar head and two tails. Is that clear? And you have two of them, and you see that they are moving, they're shaking, and you need to also be able to tell two non-polar tails, one polar head. So what does that mean? If I want to classify my plasma membrane, is it more non-polar or more polar? Remember, look at what's given to you. It has a one polar head and two non-polar tails. One polar head and two non-polar tails. So if they ask you, is the plasma membrane non-polar or polar? Excellent. It's mostly non-polar. But it is polar because the head is polar, but the rest is non-polar. Why do they call it amphipathic? Because it's two in one, polar head and two non-polar tails. So that means, you see this, I love this yellow middle big, it's like four tails facing each other, two and two, right? This big yellow structure, that's like butter, because non-polar means you dissolve in oil. So this yellow layer is butter. So what does that mean? Who's welcome to easily pass through the plasma membrane? Polar or non-polar substance? Non-polar substance. Based on the concept, likes dissolve likes. If you're non-polar, you dissolve in non-polar. If you're polar, you dissolve in polar substance. Birds of the same feather, they flock. Birds of the same feather, they flock together. So a non-polar substance will go through the plasma membrane. Give me an example of a non-polar substance that will go through the plasma membrane with ease. You apply it all the time to your skin because it goes right through the skin. What is that? What is Vaseline and lotion made up of? Polar or non-polar material? Non-polar. Because you see, cholesterol is fat. Fat and lipid and butter, these are all non-polar. So non-polar material, later on, you will learn that testosterone, ever heard of testosterone? And progesterone and estrogen, those hormones, they cross the plasma membrane with ease. Why? Because they are made of cholesterol, and cholesterol is butter. And these yellow middle non-polar tails, they will say, oh, you're one of us. You're non-polar, we're non-polar tails. But if you look like water, is water polar or non-polar? Water. Is water polar or non-polar? Water is polar. So you guys don't know what non-polar is. What polar, what is non-polar? What are you doing in lecture? People don't know what polar or non-polar is. Okay, what is polar? What is non-polar? Polar means there is an uneven distribution of charges. That's why they say there is a a positive pole and a negative pole on a battery. Is a positive charge and a negative charge. Now that's polar. Polar means what? Polar means you tilt to one side. That's why they say the South Pole and the North Pole. If you look at water, water looks like this. A huge oxygen. You know, oxygen has a molar mass of how much? 16, right? 16. What's the molar mass of hydrogen? 1. So hydrogen is too small binding to water, to oxygen. You know, water is H2, 2 hydrogens, 1 oxygen. What's the problem here? The problem here is that the bond that they have is a bond of electrons. A bond of electrons. So they're sharing electrons. Imagine you're sharing with your best friend a car. Let's say a car is $30 a month. You pay 15, your friend pays 15. You're supposed to drive the car for 15 days, your friend drives it for 15 days. That's a non-polar relationship, right? Because it's even. But if your friend is so big and bully and takes the car for 20 days and you only take it for 10, is that now the car has polarized to your friend more than you? No, that's a polar bond. So they're sharing electrons. Oxygen is sharing an electron with hydrogen. They're putting 2 electrons here, 2 electrons there. But because oxygen is so huge, it will attract the electrons to the oxygen. It will attract the electrons. The electrons are going to move away from the hydrogen and they're going to go to the big oxygen. So oxygen is going to have an inherent negative charge, while hydrogen will have an inherent positive charge. Is that clear? Is that clear? So now you see why we call this polar? Polar is when there's like a charge, like a charged particle. But if you look at carbon dioxide is CO2. Now the electrons are being shared between carbon and oxygen. But carbon and oxygen are shared because is this polar or non-polar? Can oxygen stole the electrons from little tiny hydrogen. Can oxygen steal the electrons from carbon here? No, because carbon is as big as oxygen. Carbon is like how much? 15, 14 molecular mass. So oxygen is 15, carbon is 14, 15. But with hydrogen, it's 1. So now that's an example of a non-polar bond. That's an example of a polar bond. So if you now look at the plasma membrane, you see that here you have 2 tails who are non-polar. Non-polar meaning they don't have any charge on them. But the head, what do we have on the head? We have, what molecule is that? What atom is that? What element is that at the top of the head? At the top of the polar head, what element is that makes it polar? What element makes it polar? You see it there. This element here. This one. This element here. What is it? This one. Phosphate. Phosphate with 4 oxygens. Now that's a charge. So now is water polar or non-polar? Water is polar. So water will be touching this phosphate. They are friends. Birds of the same feather flock together. Like dissolve like. Water and phosphate touch each other. But those nonpolar tails, they touch each other. So from the outside, you have water with the phosphate heads. From inside, you have water with these phosphate heads. These are the phospholipids. And you see, if you want to pass through, nonpolar substance can diffuse across the phospholipid layer. You see, the nonpolar is going to pass right through here. But the polar large molecules, they need these tunnels, these transport proteins. So this circle that is going through now, what do you think that is? Do you think that's polar or non-polar? That is polar. Excellent, because if it was non-polar, it would have went right through. So do we only look at polar, non-polar? What about size? What if you were non-polar, but you were huge? Will you still be able to go through? No, you need a transporter. And we talked about the dehydration synthesis. This is how we make the tails. You see, that's the tail of hydrocarbon. We remove water, OH, OH, we remove the OHs. And this shows us that diffusion is a process where you see what the arrow points from? The arrow points from where you have a lot of substance to where you have nothing. So here you have so many purple dots and only two here, so the diffusion pressure is to go down. That if you are non-polar. But what is facilitated diffusion? We discussed facilitated diffusion. What does that mean? Who can tell us what is facilitated diffusion? From the name, it's diffusion because the name says facilitated diffusion. So it's diffusion. But what is this whole facilitated? What does that mean? What does it mean when we say we have facilitated diffusion? Who wants to give it a shot? What's facilitated diffusion? Yes, please. So facilitated diffusion requires energy. No, that's active transport. It requires ATP. Yes, please, Giselle. It needs the facilitator. It needs somebody to bring it in because it's too big, like glucose. Glucose, if you have so much glucose outside the cell and you have no glucose inside the cell, obviously the diffusion pressure is going to be from the outside towards the inside. But can glucose cross the non-polar leaflet? It doesn't. So it needs this transporter, this facilitator, like an elevator. It literally will grab it and bring it inward. And then the active transport, that one is going to require ATP. Facilitated diffusion does not require chemical energy. It doesn't. It just needs diffusion pressure. What is diffusion pressure running on what kind of energy? Diffusion is running on which kind of energy? We talked about that last week. Diffusion is running through which kind of energy? Yes, kinetic energy. Excellent. We said the energy of bombardment, the energy of heat. It's like people coming out the subway. They push each other. They do that. They push each other to get out. That's how the energy is made. You get the energy of being pushed. But the other type of energy, ATP, that is not kinetic energy. That's not bombardment or heat energy. That's chemical energy, like burning a Bunsen burner or burning gasoline. Okay, so this is the plasma membrane, and we talked about osmosis, where in osmosis, you're gonna move only water. What is osmosis? Who could remind us what is osmosis? Yes, please, yourself. The movement of water. Osmosis is the movement of water. In which direction? High what? That's diffusion of water. That's not osmosis. Yes, please. Excellent. Because it wants to go to dissolve from a low concentration of solute, like here. There's 10% here, 20% here. Water is going to go from 10% and it's going to go over to 20%. Okay. Active transport is when we rely on ATP. Okay. So now let's look at today's lab. Today's lab is very interesting. Today's lab is very interesting. Today's lab is very interesting. Did I say that enough? Is today's lab very interesting? So let's find out what is so interesting about today's lab. Cellular membranes. So now we already talked about what our plasma membrane is all about. It is a universe. It is rich and unique in many different structures. Every structure has a function. There's protein channels in there. There's phospholipids in there. There's a nonpolar core, hydrophobic core, hydrophilic head, polar heads. Okay, so the membranes separate and organize the myriad of reactions within the cell and they allow the communication. You need to know this structure, guys. Please look at where the mouse is. This is very important. We already said that this is the structure of a phospholipid. You see, it's a two-in-one. It's a hybrid. It's amphipathic. It's amphipathic because it's two-in-one. Phospholipid. Phospholipid. You ever heard of that philosophical thing where there's like a human head in the body of a horse or something like that? That's what amphipathic is. It's like when you mix two things that don't belong with each other. Non-polar and polar never come together except here. So if you look now, you have a polar head. You guys see the polar head? And that's the non-polar tail. Polar head, non-polar tail. Polar, we said, you dissolve in water because water is polar. Water has a negative charge because it stole the electrons from who? Who did water steal the electrons from? Oh no, we talked about that, and today I'm going to be handing out zeros. I don't want to give anybody zero, but I'm sorry, you guys are pushing me. Today we're going to give a lot of zeros. The polar head is charged. The charge distribution is uneven, is not neutral. Just like water. Water stole the electrons there from the hydrogen because the oxygen was so big, it stole the electrons from the hydrogen. So phosphate is also charged. So that's why we call it a phospholipid. Now look at the structure. The structure of the head, that's the head. This whole part is the head. What is the head made up of? The head is made up of a phosphate group, a glycerol backbone, and a nitrogen charged choline. Don't worry about that. Just remember the most significant structure is the phosphate. That's what makes our head polar. Polar means you dissolve in water because water is polar also. If you look down now, that's the tail. The tail, that's the non-polar tail. The non-polar tail is hydrophobic. The non-polar tail is hydrophobic. The non-polar tail is hydrophobic because it has a long chain of carbon, 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 hydrogen. It's called a hydrocarbon. Hydrocarbon. We don't have one, we have two of those. So that's the structure of the phospholipid. This is the most majority of your plasma membrane is this phospholipid. There are proteins, there are like cell adhesion molecules, cell adhesion proteins, glycolipids, glycocalyx, lipoproteins, but it is those phospholipids that make up like the vast majority. You see now, this is the arrangement of the phospholipid in the biological membrane. And the fluid mosaic model means that what I talked about is dynamic, is fluid, meaning that is able to move, able to be dynamic, not just static, fixed like brick, like glass, like standing there. It does not move, doesn't budge. That's not true. It's a fluid. We call it the fluid mosaic model. So these chains serve as distinctive tags that identify particular cells, particular types of cells. Selectively permeable, and we talked about there's a high permeability for gases, very small uncharged molecules. So the plasma membrane is very permeable to gases because gases are too small. Gases are so tiny. That's why they are so welcome to pass through the plasma membrane. The moderate permeability is water and urea. Yes, please. Okay, so the fluid mosaic model is a theory. It's a theory that explains what the plasma membrane look like. Remember we said that the plasma membrane is like a universe and is a moving. So the phospholipids are moving, the protein channels are moving, they're constantly rearranged. Some of them are removed, some are placed again. You have some non-polar, you have aquaporins, water molecules that are hooked in the middle. You have also cell adhesion proteins. So the key here is that mosaic, mosaic is supposed to be like stiff, but here is showing you it's fluid, meaning that our plasma membrane is not stiff. It's moving, it's dynamic. There's a lot of fluidity of the plasma membrane. So because people used to think the plasma membrane was like the fence around your home. Your fence around your home is not supposed to be dynamic. It's not supposed to be fluid. It's supposed to be stiff and strong and prevent anything from leaking through it. No thief coming in the middle of the night, and no rats, and no all of that. Hopefully, they jump over it. Yeah, but this is the fluid mosaic model. It's a model. So a model always gives you a representation. A model is a simple representation. You know, when you look in the GPS and you put it on a GPS to take you home, when it tells you make a left at this street, does the street on the GPS look like the way it actually is in real life? It doesn't, because the GPS is a model. It's just a simple representation. So the fluid mosaic model, you see, the chains serve as distinctive tags that identify particular types of cells. These elaborate molecular elements, they form the fluid mosaic model. So the fluid mosaic model shows the framework and the behavior that it's how is the plasma membrane structured. And proteins may span the membrane and they may be bound on the surface of the other proteins. It's selectively permeable, and you know, this is very important because it shows you how your plasma membrane works. The plasma membrane has a high permeability for gases, very small uncharged molecules they pass through. Moderate permeability, you have water and urea. And there's low permeability, polar organic molecules, sugars. And then there is very low permeability, that's ions, charged polar molecules, and macromolecules. These are amino acids, proteins, polysaccharides, nucleic acids, DNA, and RNA. Clear? Okay, now we're going to go into the bead cells as an experimental system. So, this bead tissue will be your model. You see a simple cell. to investigate the membrane integrity. So the root of the beet, known as the beta vulgaris, contains large amount of reddish pigment called beta-cyanine. It is localized mostly entirely in the large central vacuoles of the cell. Where are the vacuoles found? Vacuoles are found only in the cytoplasm. Vacuoles are found in the cytoplasm. So in healthy cells, beta-cyanine in the vacuoles remains inside, surrounded by a membrane known as the tonoplast. The entire cell, including the vacuole, the tonoplast, and the cytoplasm, is surrounded by a cell membrane. In two procedures, you guys will subject the beet cells to a range of temperature, organic solvents, determine which treatment stress damage the membrane the most. So you know this membrane is made of phospholipids. So there, if you subject it to like hot heat, it's gonna disintegrate and burn and fall apart. So this experiment is to subject stress to the plasma membrane of the beet, and you see what the results are going to be. If the stress damages the membrane, the beta-cyanine will leak through the tonoplast and the plasma membrane. This leakage from the stress bead will color the surrounding water. The water is going to be red. That means the plasma membrane integrity has been lost, damaged. It's called necrosis. Necrosis. When the cell is membrane is damaged, is called necrosis. What is necrosis? Necrosis is the damage of plasma membrane and the leakage of all the intercellular substance. Okay, so begin. So membranes are sensitive to temperature. So high temperatures cause violent molecular collisions that degrade a membrane as a physical barrier to diffusion. And freezing temperature cause water to crystallize as ice and expand because of the hydrogen bond alignment. This expansion and formation of ice is going to rupture the membrane. So what you're going to do now is you're going to examine the treatment and state the question addressed by this procedure. So you're going to have 6 test tubes. You're going to subject them to different temperatures, and please work in groups. We have the second 10.2, that's going to be the effects of the organic solvent. So first we're going to look at the effect of temperature, then we're going to look at the effect of the organic solvent, and that's going to be it for today. Any questions? Alrighty, so there are always modifications, right? There are always modifications. And let's see if I can log into. Okay, the lecture is now over.